嗨，大家好，那我们本次的活动就正式开场喽。我是风纪生技的专员 Cassie， 感谢各位拨空参加本次由风纪主办的 Webinar 活动。在此提醒，为使活动顺利进行，各位朋友的麦克风都会保持静音。如果有问题的话，可以在问题 question 那个栏位使用文字提问。我们将在2场讲座的最后一并向主讲者提问。感谢各位的配合。那在本次 Webinar 的最后会有一个问卷连结，只要填写问卷就有机会参加抽奖活动，欢迎踊跃填写。本次活动要特别感谢 Invitae 以及 Molecular Health 公司的伙伴们，协助风纪一同促成这次的 Webinar 活动。接下来就请 Matt 为我们本次 Precision Medicine 的精准医疗主题。
嗨，大家好，我是风纪生物科技的 Matt。感谢大家今天的时间参加由风纪生物科技主办的 Webinar 活动。主题呢是 Total Solution for Precision Medicine。会议中同时邀请了 Invitae 以及 Molecular Health 共同介绍检测技术与数据分析的解决方案。在最新的 NCCN 的指引以及ESMO 的肿瘤检测建议基因清单中，总共提出了22个建议检测的基因。而这些基因中，除了典型的点突变以及音调之外呢，还包含了gene fusion 以及 exon skipping 这些重要的新的 biomarker。那这些 biomarker 呢，都跟我们现在正在使用中的肿瘤治疗有非常密切的关系。在目前现行的实验室中普遍使用的检测技术，多数都使用是一个 single gene testing 的一个做法。那这个技术中呢，需要使用包含像 qPCR 或 FISH 等不同的技术去做整合。那整合的检测的流程呢，是一个 sequential testing 的一个策略。所以对于每个病患，对于每个 case 来说呢，它通常需要耗费非常长的时间才能完成所有的基因检测。那现在在这个时间点上 ，NGS 的技术提供了一个崭新的一个思维跟一个新的策略。NGS 可以针对所有的 biomarker 进行一次性的检测。那这样的检测策略呢，可以很有效的缩短我们的检测时间。那 NGS 的技术特性呢，还可以带来更好的检测敏感度以及 biomarker 的一个覆盖性。在实验室检测中呢，它会普遍遇到了一些重要的一个议题。那 NGS 是我们现在检测中一个很棒的 solution， 它们能让我们一次完成像是 fusion 或 indel 的一个检测。在实验室中，然后自己建立 NGS 的检测流程，它会面临的2个比较首先会遇到的问题呢，就是 NGS 的检测普遍来说会比较复杂且困难。那现行的流程很多，那对于实验室来说，可能需要花费较长的时间去完成以及建立。第2个挑战呢，就是数据的分析。第一个需要处理的问题是，我们可能需要在实验室中建立 bioinformatics 的资源，才能够让我们用 NGS 的数据去这些图片的码。此外呢，如果我们要跟临床做结合，我们还需要一个是能够做 treatment decision 分析的一个报告系统，我们才能够去完成一个治疗指引的一个相关报告。那在今天的主题中呢，我们将要介绍如何整合 Invitae AMP 技术，以及它的对应的 bioinformatics 分析工具ArcherDX Unlimited 来提供这些新兴的 biomarker 的检测。那这个整合的解决方案呢，我们能够去解决我们在实验室中遇到的第一个困难，就是 NGS 的技术的建立的流程，以及整个新技术的一个开发。那第2个部分呢，我们会介绍由德国 Molecular Health 公司所开发的treatment decision 分析系统，它可以帮助我们去解决制作这种用药资讯分析报告的一个新的挑战。好，那第1个段落呢，我们会邀请美国 Invitae 公司的 speaker Kevin 以及 Igu 分享如何利用 AMP 技术进行 oncogenic 的分析。So today's webinar, we will invite Invitae speaker Aigu and Kevin to introduce the AMP technology to detect the oncogene variant. Now I will hand over to Aigu and Kevin.
All right, thank you, Matt, for the introduction. And my name is Aigu. I am the APAC marketing lead for Invitae, and I would like to give a brief introduction towards Invitae and also the recent acquisition of Archer by Invitae. Right. So, sorry. All right, and I'd like to give a disclaimer before I begin is that Invitae's kitted products is for research use only and is not meant for diagnostic procedures. And also for many of you who have heard previously of ArcherDX, we have been acquired by Invitae and is now part of the Invitae organization. And so when you hear about kitted products with the brand Invitae, yeah, we are still offering the same products in the company, right? So nothing changes. And Invitae's mission is to bring comprehensive genome genetic testing and information to be accessible to as many people around the world. So the key point is affordable healthcare and with easily accessible genetic information. And this is the mission that Invitae strives. And the acquisition of Archer is to help Invitae meet that mission by ensuring decentralized testing that is available to different countries in the world. So to get started, as a quick introduction about our core technology, we are utilizing the anchored multiplex PCR, also known as the AMP chemistry, which I will explain a bit more in my next slide, right? And we have a simple workflow for this process, and coupled with a secondary analysis, which will be shared by Kevin during his presentation later on. So the various mutations that can be detected are fusions, splicings, SNVs, indel CNVs, as well as gene expression across our multiple disease states, solid tumors, sarcomas, blood cancers, etc. And all of this can be assessed through our various product offerings. So FusionPlex, which will be used for RNA-based fusion detection. VariantPlex will be a DNA-based methodology kits that we offer, and liquid biopsy is through our LiquidPlex offering, right? So all three of these products could be used, and they can be fully customized to meet the needs of your laboratory. So to give a quick introduction about how our technology works, so the AMP technology utilizes a gene-specific primer on one end, and on the other end, it utilizes a universal primer where it doesn't have any specific GSP sequences, so it's a universal one. So it will bind to the common sequences on the other side of the gene. So the advantages of this is that we need not care about what other side gene sequences are, because that universal primer will just bind to it. And this allows for the identification, for example, of fusion genes that otherwise could not be identified if we were to hinge the other end of the PCR primer, right? So that it only binds to sites that we know of. So the other part about the AMP technology is that it comes with a molecular barcode, which allows us to do the bioinformatics processing easier later on downstream. So one of the key things is that the molecular barcode allows us to effectively remove the noise and also help in advanced error correction, as well as give unique countings to eliminate problems caused by PCR related artifacts or duplications. And finally, our workflow is relatively simple. So in terms of the steps, all of the various assays go through similar processing. So you take the sample input, and then we do the adapter ligations as well, coupled by the 2 rounds of PCR, right? The only difference between RNA and DNA is that you will first have to do the reverse transcription before you get that cDNA and before proceeding. At the end, you are then able to load all of these various assays, if you are working on DNA and RNA at the same time, and then add them onto the sequencer and run it through the Archer analysis platform. Okay, so I'll now play a recording that Kevin had. So my apologies that Kevin is not able to attend the call today due to the challenges in the time zone. It's hard to accommodate the US and our time zone as well as European time zone. So unfortunately, Kevin is not able to make it today, but he will be able to help answer any of your bioinformatics related questions to Archer analysis. So please feel free to share your questions and we will answer it for you.
First, I'd like to talk about the purpose-built analysis platform called Archer Analysis that we have to offer. So Archer Analysis is a custom built from the ground up to take advantage of the unique elements of the anchored multiplex PCR has to offer. So as soon as the sequencer is complete, the analysis will kick off, taking a FASTQ file through our powerful bioinformatics pipeline to result in advanced visualizations, variant calling, and reporting. Analysis is built for all levels of users with a friendly point-and-click interface to provide deep analytical metrics for power users, yet simple tiered visualizations and filters for basic users. The platform can be implemented locally on your server, behind your firewall, or a private, secure cloud instance. A robust API enables integration of Archer Analysis into a broader workflow, including a LIMS system and third-party clinical interpretation and reporting tools, such as Molecular Health tertiary analysis software. So from an Archer Analysis standpoint, we can deploy, interpret, and report. Next, I'd like to talk about our adapters that enable our error correction and deduplication process. So in the top two figures, you see our unique error corrected reads. So in the upper left hand corner, you see our raw reads that are then error corrected and then deduplicated into error corrected reads. So the error correction process and deduplication process happen at the exact same time. So in the figure or in the visualization with the yellow box, you can see a list of raw fragments with the same molecular barcode associated with them. And in each of those fragments, you can kind of see sporadic PCR sequencing errors that might be occurring, as well as a true mutation with that T-SNP there. So what happens is we can error correct and deduplicate those reads into one consensus molecule that you can observe underneath that red arrow. So here we've corrected out the PCR and sequencing errors to create a more robust read to be analyzed. So you can kind of see how we go from the raw reads to the error corrected reads and how much cleaner it all looks. So from the deduplication process, the figure on the bottom here is a nice visual where it shows individual molecular barcodes that are ligated to each of the raw reads. So these raw reads are the total number of reads or read pairs reported from the sequencer. Then we deduplicate those into unique fragments or molecular bins. So this is the total number of unique fragments extracted from your sample. These are determined by finding all the raw reads or read pairs that share the same molecular barcode and deduplicating these into a single consensus sequence. Then we take the unique fragments or molecular bins kind of a step forward and assess the unique start sites as well. So the unique start sites is a subset of the unique fragments and represents the total number of unique fragment lengths extracted from your sample. Since each fragment is anchored by a GSP2 on one side, lengths can only be affected by the random start sites on the opposite end of the fragment. So unique start sites can manifest as multiple physical molecules, which is why there are typically more unique fragments than unique start sites. So unique fragments tell the true input quantity from your sample and allow for accurate detection of allele fractions. Unique fragments and unique start sites together provide a reliable measure of sample complexity, which can be correlated to the assay sensitivity. These metrics also help guard potential false positives arising from PCR and sequencing error. Next, I'd like to talk about some of the key features Archer Analysis has to offer. This screenshot shows how simple it is, of a platform it is, and how users can set up a job by selecting the desired pipelines and dependency files to be used to analyze your samples. So Archer Analysis, again, can be installed as a local or cloud installation, so a VM behind your institution's firewall, or a private, secure instance with maximum scalability and ease of use. We also have a user interface that's easy to use, web-based portal, where no programming experience is required. Archer Analysis also can be automated by using watch folders and API, which I'll get into in just a little bit. You can also use organization management, which is advanced permission management for organizational collaboration. So watch folders is a mechanism that allows the automatic execution of a predefined workflow whenever a set of BAMs or FASTQ files are moved to a specific watch directory. Now there's two steps in setting up this workflow. You first have to set up the watch folder on the user interface, which is simple to do, and also develop a simple script to move your FASTQ or BAM files into a watch directory. So what happens is once those files are deposited once those sequencing files are deposited into those watch directories, software can automatically be pointed to that, extract the files, and begin processing them. We also have a REST API supported by Swagger. So this can be used to automate or customize your analysis and integrate into third-party or LIMS systems. REST API can automate the processing of jobs post-sequencing as well. Archer Analysis's REST API is self-documenting and supported by Swagger. And Swagger is a powerful and easy-to-use tool for all developers that it can be used to help them throughout the entire development process. So automating Archer Analysis, when you can leverage both watch folders and REST API, which can allow you to automate your workflow post-sequencing all the way through integration to a third-party interpretation and reporting solution. So with the proper implementation of scripting, output files generated by Archer Analysis can be automatically pulled into a tertiary analysis tool. Next, I'd like to talk about detected fusions/mutations by Archer Analysis. So some examples I want to go over today are an ALK fusion example, a ROS1 fusion, an FGFR3 fusion, a MET exon 14 skipping event, and an EGFR point mutation. So here's an example of a non-small cell lung cancer sample that harbors an EML4 ALK fusion event. This is an example screenshot of our fusion grid coming from our user interface showing the strong event. You know, in order for an event to be deemed strong within our fusion grid, there are certain strong evidence criteria that needs to be met. The event has to have greater than or equal to 5 unique breakpoints spanning the reads. It has to have greater than or equal to 3 unique start sites and greater than or equal to 10% of the reads supporting the event. With the caveat that if these aren't met, if these thresholds aren't met, but this event is known to our Quiver database, it will be shown to the user as a strong event. Now, the Quiver Fusion database is the largest curated database of known fusion events. So it indicates the known status, whether it be exact breakpoint known or partners known and their breakpoint as well. And you can also link out to the publications of those fusion events that are located in our Quiver database. So as you can see here in this screenshot, there's an example of a bullseye with the exact breakpoint known, which means this is an exact breakpoint into our Quiver database. And what it does is it just gives the user confidence that this mutation has clinical significance and the publications to back it up. Here's an ALK visualization, ALK fusion visualization in the JBrowse visualization tool that we use within Archer analysis. So it's a nice consensus view of the fusion event, where you can see with the red line going on the screen the EML4 fusion occurring. So in this visualization, not only are we showing the bed annotations of the fusion events, but we're also showing the bed GSP2s at the top that are contributing to the fusion event, as well as the VAM coverage and the reads contributing to this event to help for visual inspection of these mutations to add confidence in your fusion calls. Next, here's another non-small cell lung cancer example, which harbors an EGFR3 and a TACC3, as well as a EZR and ROS1 fusion events. So again, very similar to what I just explained, these mutations here are being reported as strong events because they hit the criteria that I just discussed, where you need to have greater than or equal to 3 start sites, greater than or equal to 5 reads, and greater than or equal to 10% reads. Not only is the evidence strong for both of these mutations here, but they're also both known to, with exact breakpoints known to the Quiver database, with the bullseye indicated in the icons under the filter in the visualization. The visualization is also a great tool to kind of conceptualize what the fusion event looks like. And this visualization is surrounding the breakpoint that is being displayed in the fusion grid here. Along with these fusion events, we also categorize it as a fusion or oncogenic isoform or novel isoform. And we also can determine whether or not the mutations in frame. You can also observe that there are the TO and report columns on the far right side. The TO, or total observations, is also a great tool or great metric to also add confidence to a specific fusion event or mutation that's being called. Now, the total observation are the amount of times this specific mutation has been observed within your instance of Archer analysis. And here are the visualizations for both the EGFR3 and ROS1 visualizations within JBrowse. Here you can see the fusion events for the 2 genes, as well as the evidence that is supporting the mutation that's being called here. Next, I'd like to show a MET exon 14 skipping example. So just to kind of explain what it is, so in some cancers, exon 14 of MET gene is skipped, resulting in the joining of exon 13 and exon 15. So MET exon 14 is an oncogenic driver in non-small cell lung cancer. And again, this is familiar to you all. This is the fusion grid that is reporting out this oncogenic isoform of this MET exon 14 skipping event, meaning the 3 start sites, the 5 reads, and the 10% reads criteria. And again, this is also a known breakpoint to the Archer database. Here's the visualization of it, where you can see the MET exon 13 fusing to the MET exon 15, and MET exon 14 being absent from this consensus view of this oncogenic isoform. And the nice support of the BAM coverage, as well as the read alignments supporting this event. And the last example I wanted to just shed some light on is this EGFR point mutation and the visualization within JBrowse. So with our point mutations, there are specific strong evidence criteria that needs to be met in order for the mutation to be considered a strong SNP or INDO. And that has to have greater than or equal to 5 reads or alternative observations, 3 or greater unique start sites or unique alternative observations, an allelic frequency that is greater than or equal to 2.7%. It has sample strand bias is a no, and has a consequence that is reported by ensemble of that. Now on the right-hand side of the screen here, you can see the visualization within JBrowse, where the top track you have the gene or the EGFR. You also have the variant ROI or the region of interest that the software is interrogating, as well as a targeted mutations list that is being used to detect this point mutation here. Now, targeted mutation files are very important. So when using the RNA or DNA SNP end-up pipelines, the use of a target mutation file is recommended. So if there is a specific mutation of interest when running the SNP end-up pipeline for either DNA or RNA panels, then a list can be compiled in a variant call format of VCF that can be supplied to the software before processing a batch of samples. So this will force the software to report on each mutation in the list, regardless of being called by the pipeline. So using a targeted mutation file with a SNP end-up pipeline is an excellent way to surface information on mutations of known significance for your application. It can be used simultaneously, searching for non-targeted variants so that no stone is left unturned. And lastly, the reporting capabilities of Archer analysis. So within the secondary analysis, you can create a customizable job report and sample summary report. Or you can use our robust REST API and PostScript hooks to integrate with a LIMS or third-party interpretation and reporting tool. And here's some screenshots that kind of give an example of what the secondary analysis report will look like by default. But with competent knowledge in HTML and our REST API, developers can create their own custom report to upload to the secondary analysis to generate reports moving forward. And that's all I have.
All right. Yep. So thank you, Kevin, for a presentation, and yeah, handing this back over to you, Matt.
谢谢Kevin 的精彩的介绍。那我们从刚才 Kevin 的介绍中呢，我们可以透过Invitae 的 AMP 的技术来取得基因检测的 variant 数据。那接下来呢，我们该如何将基因检测的数据转换成临床可以使用的 treatment decision 的 report 呢？那接下来我们欢迎 Molecular Health 的 Dr. Peter Koch 来为我们介绍如何利用 MH Guide system 来制作一个与用药相关的一个分析报告。那我就 hand over the presentation to Peter Koch to present the analysis using Molecular Health Guide.
Thank you very much, Matt. Thank you very much for the kind introduction. And I would like to take the opportunity to thank the organizers for inviting me and giving me the chance to present the MH Guide software to this audience here today. So as the third part of today's session, it's all about bioinformatics and about the implementation and converting the knowledge that is gathered from cancer genomic profiling by MH Guide into treatment decision supports based on Invitae NGS data. And I've organized my talk in 4 topics, so to say. I will give a very brief introduction to molecular health. Who actually are we? Then I will present the MH VCF adapter suite, which is a necessary interface in order to implement the NGS data that is coming from the Archer Invitae systems. And the heart and core of the talk naturally is the MH Guide software itself, with the peak of the pyramid, which is the report. And finally, of course, a summary of all important take-home messages. So who is Molecular Health? We are a bioinformatic company. We are located in Germany, Heidelberg, with a small franchise also in the US as a very important market in precision medicine. We are a medium-sized enterprise with around 115 employees. And the focus of the company is, as already mentioned, bioinformatics. So Molecular Health has a strong experience and expertise background in capturing data and big data approach in order to match the needs for precision medicine, that's somatic workflows, and in extent also germline interpretation, which however is not the major focus of today's talk. However, it's absolutely important to say that MH-Guide is registered as an in vitro diagnostics or medical device in Europe, that is, and that's coming along with prominent certifications, the ISO 13485, also kept CLIA accreditation in order to assure that the quality that is provided in treatment findings based on NGS indeed is suitable for a clinical approach. And this is the point. The IVD, of course, is then the next step over research use only applications, as it's designed for clinical applications. And for a user perspective or end user perspective, it's also most important to mention that as we are located in Germany, also Molecular Health, we have our own IT infrastructure, which is maintaining the security in data transmission, or mainly also in the privacy of the data that is required for analysis in this background. So I'm talking about the Molecular Health Guide software, which is a made in Germany premium product for treatment decision support. So here you can see the basic workflow of any NGS analysis, and this is also being shown in the previous talks. However, the concept is most important to make clear. So the first step naturally is the NGS laboratory or the wet lab part. Again, this is what Molecular Health is not doing. However, the outcome always is a FastQ, as in raw data, which is funneled into a bioinformatic approach. And this is where Molecular Health is operating. So the second block is what MH-Guide is doing. In alignment, the FastQ is converted to a binary alignment file and reference genome alignment. And finally, the outcome of a bioinformatic pipeline always is structured data, which usually is VCF or similar structured files. And here in step 3 and 4, this is actually the major focus of my talk, which is clinical annotation, because sequencing, of course, is very important. It's the initial step in any analysis that you do. However, you need to convert the findings, as in genetic variants, also into clinical use, into actionable use, into treatment options. And this is what we refer to as clinical annotation, and this is exactly what MH-Guide is doing, which is resulting in a brief and concise report with treatment options for individual cancer patients. However, there's an interfacing structure required, as the VCF outcomes that are produced from each pipeline are different. So there's an adjustment being required, and that's always depends on the pipeline that is being utilized up front. And that's why Molecular Health have established and created the Molecular Health or MH-VCF Adapter Suite, which is readily available to our customers. You can easily download. It's from our systems, and it's a Java application, as you can see, and it's most easy and straightforward. You just go to the Invitae platform, where you obtain your NGS results, which have been nicely presented by Kevin up front. You download those files that you require and simply drag and drop or upload them into the MH Adapter Suite, which is DNA workflows, RNA workflows, structural variants such as CNVs or exon skipping data, and you just push the button to convert those Archer outcome files, and you are presented with a VCF that can be readily implemented in the MH-Guide software. So now I'm going to the MH-Guide software itself. It's time to get started here. So you can see on this overview page how the rest of my talk is structured and organized, because MH-Guide is an expert tool in treatment decision support that's organized in different perspectives. So most important, of course, is here the last box. This is the report generation. This is the clinical focus, which is provided as PDF readily to be used for the user, but also in structured formats and bioinformatic formats such as JSON or XML. However, while this is the quick way to extract the most important information, also expert perspectives are being provided. And this is most importantly, of course, the entry point, the next generation sequencing perspectives. There's a focus on medically important variants and also pharmacological perspectives, which are complemented with further expert knowledge. And this is what I would like to do with you right now. I would like to walk through those different perspectives and finally ending up in the summary with the report. So this is the entry point. This is the genetic perspective. And here you can see a list of the variants that have been identified, and these are the very same variants that Kevin also has focused on in the previous talk. Here you can see several fusions are being listed: a ROS fusion, ALK EML4, the FGFR3 fusion, a MET exon skipping event is included, and also an EGF receptor SNV, a single nucleotide variant. So obviously I'm focusing on a lung cancer case here. And in this entry point, in the initial perspective, there are several major questions that are asked by MH Guide. So first of all, what are the variants that have been identified in total? Which of them are reliable in order to associate them with medical treatment options? And this is what you can see here already with the intuitive traffic light color coding systems will be better visible in the report, as in effective treatment options. Ineffective ones are resistance mechanisms, and safety biomarkers would highlight adverse events, which however are not included in this panel analysis here. In extension of this expert perspective, you can also see that each variant is provided with further expert knowledge, such as outlinks to Ensemble or RefSeq transcript coding serogenomic positions. And to be strictly speaking, beyond the somatic perspective, also a human geneticist perspective is included for DNA variants, that is by ACMG classification and much more. So most importantly, also you can see here that each variant is accompanied with variant detailed information, such as the variant allele frequency, if we're coming from a DNA workflow observation quality coverage. These are NGS parameters. There's outlinks to prominent or connection to prominent databases in the cancer background and population frequencies, which give a strong hint whether we have a germline or a somatic variant. And this is a very important point. Lineage discrimination can be very important in cancer predisposition, which can be also highlighted by MH Guide. So here we can see the next perspective, which is CVIs, because in the end you need to convert the knowledge into actionable data. And this is what MH Guide is doing in this CVIs tab. Here, there's a focus on all the medically relevant variants that have been highlighted with a medical impact. And this is matching the finding that are coming from the patient with the curation from Molecular Health internal system. And MH Guide is continuously updated. It's updated on a monthly basis or even more with the variant information. This is redundant to the previous perspective. And most important also is that biomarker evidence levels are being included. So this is a very obvious biomarker. ALK EML4 is very prominent in NSCLC. So here you can see that different evidence resources are implemented. This green button is reflecting the ASCO tiers with the highest level. There is a Molecular Health internal ranking, which is the number 7, is also here referring to an approved biomarker with approved treatment options. And finally, this 3 marking system is FDA evidence, also highlighting the highest evidence level. And to complete the entire picture, we have the variant perspective, what has been identified. We have the biomarker evidence levels, and of course also the respective treatment options are being listed here that are coming along with this effective impact. So here you can see then, as a next perspective, I'm focusing on the pharmacological perspective. These are effective treatment findings, and of course I'm only showing one snapshot here. There's so much knowledge being implemented. Just to show you this as an example, of course, in the pharmacological perspective, the treatment option is at the center focus, which is coming along with drug approval states, and those are matched nationally specific. So here you can match this drug approval state to whatever country you're interested in, coming along also with guideline information, and in theory also pathways of the impact for this biomarker and treatment option can be highlighted at this point. Clinical trials are also very important, and this is the next step, as effective treatment findings are being listed here in the context. So in this case, it's gefitinib, are listed in the context of clinical trials. And in this case, I can show you that 3 different countries are being matched to these findings, which is China, Taiwan, and the US. And it's really impressive that for those 5 variants that I've shown you, more than 130 clinical trials have been found in total. So as a next step, there is ineffectivity and safety treatment options, which again is a pharmacological perspective. And this is very interesting and important, since MH Guide is highlighting you also resistance mechanisms overall, and beyond that, even conflicting evidence is highlighted. So here we can see that based on those two different variant findings, there's differential impact being highlighted for this particular treatment option, alectinib. A conflicting evidence or resistance mechanism has been shown, and you're given the chance to resolve this conflict, while also the report will directly warn you about such a finding. So this is a very interesting feature in general, also with those toggle switches that are provided on each and every of those different perspectives. You can design and compile the evidence and findings that you want to show up in your report in order to share it with a colleague, a patient, or even in a molecular tumor board. This is when I'm coming to the report. This is maybe the most important piece. Finally, of course, again, you can either use MH Guide end-to-end, directly go to the report and save time, or you can consult the expert perspectives. And here you can see how this exemplary report for FCB in collaboration has been designed. And it's absolutely necessary to mention that MH Guide report templates are customizable. So you can, for example, insert your own logo or compile the complexity and depth of findings and detailed information that you want to have. Reports in general are organized with general information with respect to the analysis, what kind of analysis or lab test or panel assay was being conducted, and some general information, of course, also about the cancer entity and the patient are being required. To go on with this, then there is a list and overview of the main detected variants. And again, this is resembling the findings that were presented by Kevin. Here we have an overview about the RNA findings. So you can see the biomarker, you can see the exon location, what exons are being affected in those fusion genes here, for example, with the exact breakpoints already. Also, along with the evidence levels for the biomarker and also in consultation with unique reads and the percentage of reads that are being affected here. For DNA variants, it looks a bit different. There's also a very concise detail that I've shown on the variants tab. However, here, of course, we are referring to also the protein location, also the variant allele frequency, which is slightly different as a parameter in display in comparison to the RNA reads. Most importantly, the MH Guide report is giving you an overview about all the treatment findings. And here again, you can see the most intuitive traffic light color code. You can see that 36 effective treatments have been identified in total. You can see the resistance mechanism for the alectinib. The software is directly warning you about such conflicting information, which means that you, as a user, can directly go to the report and take the results as reliable, while the left-hand side of the table is focusing on the drug and treatment information, impact, and also drug approval state. And the right-hand side of the report is focusing on the biomarker information and the biomarker evidence level scores, while also clinical trials are on display. And again, I can only repeat, this is very impressive in the amount of findings here. So in total, there's a lot more information on display that I am not showing you today due to the limitation of this talk. However, all those treatment options are elucidated in further detail and providing also summary narratives that come from the molecular health curation team, and also with PubMed outlinks that can be consulted in order to study the primary literature. And this is when I come to the end of my talk. Again, I would like to thank the organizers for giving me the chance to present today. I've shown to you today the MH Guide software, which is a made in Germany premium device, which is registered as an IVD in Europe with a clear intention as treatment decision support tool in order to annotate next generation sequencing findings with a major goal to identify treatment options for individual cancer patients. MH Guide can be utilized to analyze a gene panel of your choice. So any panel, if it's customized or from a commercial provider, can be analyzed, and that's including liquid biopsy also, and any cancer entity that you're interested in. The MH Guide report is provided as PDF or also in structured formats in bioinformatic formats, and is providing not only the clinical perspective, but also expert views with full transparency to the data, and also modification of the report is possible. And also most important is to mention that MH Guide is routinely used in expert panels and molecular tumor boards as we are speaking here. And then I've highlighted, and maybe this is the most important statement of the entire session today, that I'm very happy that FCB, Invitae, and Molecular Health have joined together in a collaboration in order to establish a seamless workflow to readily and easily and directly analyze next generation sequencing data from Invitae Archer panels in the Taiwanese market. And with this final statement, I would also like to close my talk. I would like to take the chance and also thank the entire Molecular Health team for the continuous work and support in providing this expert tool and others beyond that. And also, of course, I would like to thank everybody here in the audience for your attention. Thank you very much.